Um, let's go ahead and get started. There are a couple of things that I wanted to make sure to mention, and then we'll spend the rest of today talking about the paper. So, first mention, if you did not see last night, I sent an email. It's an announcement. I'd like your feedback on the course. If so many people do it, there's extra credit points in for the whole class. So, please feel free to do that. I'm not forcing anyone to do it though. Second thing, yes, we still have today and technically 2 more lectures to cover before Friday. We're obviously not going to do that, and that's okay. So here's what we're going to do. The 2 listed as microbial communities, we're actually going to toss them out completely, so don't even open those, don't even do homework from them, yada yada yada. What we're going to do Wednesday, there's a new paper that just came out, and when I mean just came out, I mean like last week Wednesday, that says Staphylococcus epidermidis produces a chemical that actually protects us from skin cancer. I want that to be our concept for the day to discuss microbial communities. And we'll talk about the benefit of having some of these microbes. So I'm going to take some of those concepts that we would be talking about. For example, how does the skin select for types of microorganisms and not allow for the growth of others? We'll take some of those concepts and we'll apply it to this particular example. And I think it'll be nice to have something that's so fresh and new to talk about. So the homework for Wednesday is technically there is nothing to do. But what you'll see is this is going to pop up as an announcement around 1 o'clock today. It'll also, it may be emailed to you as well. I'll check to make sure that it does. But there's going to be 3 links to different articles. They largely say the same thing, but you may want to look at more than one of them. I would say take a look at a couple so you understand what we're going to be talking about. And then for Wednesday, what we'll be doing is I'll be grading participation points for the day. So, participate, in other words, is what I'm asking you to do. Beyond that, Wednesday, Julia has review, and then I will post a little bit later today when I'll be around during this week with extra time if you want to talk about stuff on the exam. Okay? But I wanted to take a few minutes now to cover what to expect on this exam. There's not a lot in some ways. This is kind of a quick turnaround, and I realize that, and next year I'll be making changes to when the first exam and second exam are to accommodate that. But I wanted to just take a few minutes to kind of highlight what we've talked about and then kind of point yourself to areas to prep for Friday. All right, so in terms of, and please ask questions if you have them as I'm going through this. In terms of microbial growth, we've got 6 objectives. Identify each of the 4 phases of growth, so lag, log, or exponential, stationary, and death. Be able to describe. Can you, in a sentence, summarize what's happening in each of these phases, or would you be able to identify which phase it is from a description? Okay. Second, classify the organisms based on optimal growth conditions. This is kind of the majority of what we have done with this section. So, what is the difference between a mesophile and a thermophile and a hyperthermophile, so on and so forth. Same thing with salt concentrations and pH concentrations. Also, with oxygen requirements, explain how oxygen affects the growth. So we talked about aerobes, or obligate aerobes versus obligate anaerobes, and not only does that affect the location in a thioglycolate tube or environments that they would grow, but it also has implications in terms of what we can predict about their metabolism. So being able to correlate whether an organism would be predicted to perform aerobic respiration or anaerobic respiration or fermentation based on where it grows in a thioglycolate tube is something that I would like you to be able to do. Know the structure and function of spores, Largely here we've talked about spores in the context of being quite resistant to environmental stressors, being resistant to chemical and also to physical methods of growth control. And so that's the big concept there. We've talked about methods to measure growth, and we've done 2 of them in lab as well. So spectrophotometers as an indirect method, plate counts and sterile dilutions as direct method, and then finally calculate and evaluate growth. There will be a calculation problem on the exam, so bring your calculators with you. You may even have to analyze some data and interpret it, kind of like the spore problem that we did on Friday, where we took a look at some data and said, what are the implications? Why does isogel, for example, in Purell not work in inhibiting Clostridium difficile spores, but hand washing does do the job in removing them? Okay. Questions on those objectives? All right. For the next set, bless you, control of growth. Describe physical and chemical controls that are commonly used to control growth. The physical controls, we spent some time talking about the different types of methods, so you should be able to describe the methodology of pasteurization, filtration, why heat, or how you might apply heat to kill a microbe, how acid might work to inhibit a microbe, so on and so forth. We didn't really talk about specific chemicals, right? But we did talk about ways that we can analyze and determine which chemical is more effective than the other. And we said, we talked a lot about how can we group chemicals into categories of sterilants, disinfectants, antiseptics, or degerming agents based on some key characteristics. Okay, and that's what I would like you to be able to do with that information. I suppose in the same way, distinguish those terms from each other. What makes a sterilant different from a disinfectant, or a disinfectant different from an antiseptic? Develop strategies and then analyze data. And this kind of goes along with what we talked about last week on Monday and Friday, where we measured zones of inhibition, talked about minimal inhibitory concentrations calculated by the broth dilution assays, and how we might make a decision as to which of the 2 chemicals or 3 chemicals is the best at inhibiting a microorganism. Questions there? Okay. For the last set, for this microbial growth one, this may be in flux, so I'll let you know if things change. But right now, explain how environment can influence microbes. This actually fits with what we've talked about, in all honesty. So for example, we'll talk on Wednesday the microenvironment of the skin and how that selects for the growth of certain types of microorganisms and inhibits others. So that ties together back to growth and growth control. This will be a great example of how what we call commensal microorganisms, or those that don't cause harm to us, can actually be beneficial to our health. And 7.3 might get crossed out, know how they're identified. I don't know if in this paper or in the literature they talk about how did they identify staph epi. My guess is no, so this one will likely be crossed out. And the last one definitely is crossed out, since that's not something we'll cover on Wednesday. So it's highly focused. I think that the videos are, like I said before, are a great study tool for this and reviewing some of the concepts. There are also a couple of exams available. This is one area where in the past there's been some deviation in what I've taught from one semester to the next. So you're going to note that for this this unit, there are actually 2 sets of tests loaded up on the Blackboard. There are a couple listed as test 2. There are a couple listed as test 3. Make sure that the information you are using to test yourself on those exams is the information that we've talked about. There are certain things like antibiotics, DNA replication, etc., that we have not discussed yet, nor do I expect you to know that material for this exam. That comes after spring break. We're going to talk about this paper that I had you read overnight, but I want to break into groups of 3. So break into groups of 3 for me. Wait, let me see how many we have here before I say that. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Ah, crap. 2 to 3. Break into groups of 2 to 3. So what I'm going to do is I'm just going to hand out one question for each group of people. And what I want you to do with your partner is answer that question to the best of your ability. I'm going to give you about 5, maybe 10 minutes to work through this. These are from different images of the paper. And then what we're going to do is do a little kind of jigsaw almost, where I will have you report out on this so that we can talk about the paper as yours. You've got a head start on that one. All right. So get discussing. I will come by and answer any questions that you have, but this will be a great place to start our conversation. Right, so determine whether soaps with triclosan actually have a benefit in removing bacteria during the hand washing process, right? Now, you could maybe understand the hypothesis from historical, or you could just take that purpose and turn it into a hypothesis statement. That's the easier way. So the hypothesis would be triclosan, or hand soaps with triclosan do a better job of inhibiting microbial growth on hands, or do a better job of killing bacteria than does regular soap and water.
I guess when I was reading it, I didn't really feel like that was necessarily what they were actually hypothesizing. It almost seems like it came in more with a bias that they like that because of this FDA thing, like that you have to actually prove that it is useful, or that the benefits outweigh the risk, or that it's useful in a real life use.
Yeah.
I guess when I read it, I didn't get a clear sense that it almost seems like they were testing to confirm that it doesn't.
I think, and yes, is the answer to that. I think their bias is that it doesn't do anything. And I think that becomes kind of clear in the way that they set up the introduction in particular. So if you read the introduction, there's parts like, here are the negative side effects of triclosan. And it talks about things like disruption of hormone levels, and some people are sensitive to it on their skin. And there's a number of issues that they list. I think if you're looking for a broad hypothesis, and again, a hypothesis, in the end of the day, it doesn't matter if you are correct or if you're incorrect. In other words, it doesn't matter if your results support or refute your hypothesis. Just throw something out there. Right? They could hypothesize triclosan will do nothing. They could hypothesize triclosan is a godsend. It will improve the quality of hand soaps. Does it change the end? Does it necessarily change the setup of the experimentation, so the techniques that they're using, or does it change the interpretations that they make at the end? Not really, right? So, but I completely get what you're saying. I think those are really valid points, and it shows a deep level of reading the material. So thanks for sharing that. If we were to do this, now think about, we kind of talked about this on Friday, I think. How do we wash hands? In the United States, how are we taught to wash hands? Under warm running water with soap. For how long?
20 seconds.
Okay. Which of the experiments in this study actually fits that? There's 4 figures that they look at, constituting 3 different experiments. Which of the ones actually test that, Alana?
Figure 2. The one
You're right that does. Absolutely right. Which maybe is, and I did not think of it that way, but that definitely is true. So they test in 1 versus 2, room temperature water versus warm water to see if there's a difference in effect. How about on actual hands? 4 they use for that. And the other thing to think about is time. How long are we supposed to wash hands for? 20 seconds? I don't want to jump ahead, but if you want to take a quick look at figure 3, look at the time at which triclosan is having an effect. Okay, we'll come back to that. So to test the hand washing hypothesis, really it's only figure 4 that's testing it. These other ones are all kind of modifications of that experiment. Right. So let's go ahead and let's take a look at the setup. Let's take a look at, that's not what I wanted to do. Let's take a look at figure 1. All right, so who had the diagram, the setup for this experiment? Kelly, do you guys want to just describe what they're doing?
So the 2 graphs, the x-axis for the first one is the gram-positive bacteria, and the y-axis is gram-negative bacteria.
Okay.
And then the y-axis for both of them is measuring like the bacterial population in general, and then each of the 3 like bars, whatever the white bars are, the population of bacteria, and then comparing the use of plain soap with the.
Good. How do they do this experiment? So in other words, tell me a little bit about the methodology. What do they do? Are they doing this on hands? No. I think it's pretty much test tubes. Take soap, add bacteria to a test tube, measure concentration before, measure concentration after, what is it? Short period of time? Yeah, it's in the seconds range. Recall one of the things that we mentioned about a scale bar like this on the y-axis here, this is in log CFU per mil. So the key point to remember here is that we can just say 10 to that number. So in other words, the measurements in terms of actual CFU per mil is 1, 10, 100, 1,000, 10,000, 100,000, so on and so forth. Okay, so those are 10. Each of those numbers represents a tenfold change in the amount of bacteria. If we were to draw conclusions, or let's not even draw conclusions yet. Let's just analyze the data for one of these. So would somebody be willing to talk about the effects of plain soap and antibacterial soap on Enterococcus faecalis 19433? The first one up there. Molly, go ahead.
Population with plain soap having slightly less.
Okay. How much of a reduction?
So like I don't know, like what would you call 10? What?
Yeah, it's a good question. How do we actually say this, right? There's a couple of ways that we could say it. One, we could say there's a 1 log reduction. That would be the simple way to say it. If you wanted to convert this to out of the logs and into like whole numbers, you could say this is a tenfold difference. In other words, 90% decrease or a tenfold reduction in the amount of bacteria after washing. Now, you pointed out that, yeah, there's some difference between the antibacterial soap and the plain soap. Do we consider this to be a significant difference, though? Lana, you're saying no. Why not?
I don't think it's a big enough difference.
I would agree with that. Usually in the bacterial world, we're oftentimes concerned with tenfold or greater differences. So if we really wanted to see the triclosan has an effect, we'd want to see this bar like down at 4 or 3. That would be one way to look at it. What's another thing we could look for, Taylor?
Do they only do each of these tests?
That's a great question. How many times do they do it? Does it say? Is it 3 times in there? So we would want replicate experiments, right? The error bars suggest that there's some sort of replication, but I don't remember off the top of my head if it's 2 or 3 times that they do this experiment. Why do you bring that up, though?
One test could like throw off the entire data. So if they have like one really good test and
Yeah, and that's why we do the replications, right? To kind of tease out whether there are artificial results like that. We also can look at, well, what do these represent? This line right here. What does that represent? Standard deviation, or an idea of how different are those results from one experiment to the next. How can we use that to determine if there's significance in this data? So for example, are these different or the same? Would we consider these to be different or the same values? Or statistically different? One simple way to look at this is look to see if those error bars overlap with each other. If they do, you can say there's not going to be a statistical difference in the data. In other words, we could say the two of them, just regular plain hand soap versus hand soap with triclosan, they work equally well. That's one conclusion we can make. And if we look at all of this data together, what is the general trend for those gram-positive bacteria? Is there a difference between soap with triclosan and soap without triclosan? Not really. There's differences between the bacteria on how they're affected, but if we just compare those two different soaps to each other, they look pretty similar in their effectiveness. We see the same thing with gram-negative, so I'm just going to skip beyond those right now. But when we begin to look at figure 2, now what's the difference between figure 2 and figure 1? And Alana, you already pointed this out. What's the difference between figure 2 and 1 here? Temperature. This one's done at 22. Figure 2 is done at 40 degrees Celsius. So let's take a look at, see if I have this set up the way I want to in Excel, or in PowerPoint. Nope, of course I don't. Why would I? Bear with me for a minute. Nope, that's not what I want. Okay, top one here represents gram-negatives at 22 degrees Celsius, so the figure from, or the image from figure 1. Bottom one here represents the killing at 40 degrees Celsius. So compare these results to each other. And what I want you to do first is just turn to your neighbor and tell them, is there a difference? Does temperature have an effect on the ability of these soaps to inhibit microbial growth? Who thinks there is no difference? What evidence are you using to support that decision? So what evidence do we use to say that there is no difference? Molly, go ahead.
I mean, if you just look at the whole population for both of them, they're both on the stage, I guess. Just comparing
Yeah, no, where they show up, their y-axis values are similar, right? So if we were actually to superimpose these two images on top of each other, they'd almost look identical. Actually, I had to do a double take to make sure I didn't copy and paste the same image. And they look very similar. There's very minor differences. But overall, what we're looking for here is what's the log reduction. And in each of those cases, for each of the bacteria, like for example, Campylobacter jejuni, and the top graph at 22 versus the bottom graph at 40, there's maybe a log reduction in each case. It's somewhere between no change and a log reduction. And we see that throughout all of those that are being measured, that the two images look very similar to one another. Because there's no difference there, our interpretation would be, at least in the way that they've performed this experiment, temperature has no effect whatsoever. Okay? Based on this, who uses warm water to wash hands typically? Would you change your practices? Would you say, it doesn't matter? Molly, go ahead.
Well, I was always taught when I started working in healthcare not to wash with warm water because to get to the heat that you would need to actually have an effect, you wouldn't be able to like tolerate that. And the negative effect is that like overuse of hot water creates cracks in your skin, and you're more likely to like get cuts, which are riskier to exposure.
Yeah, it creates a break in what we would call the first line of defense, skin, which is a huge organ that prevents disease and infection. That's a really good point. 40 degrees Celsius supports the growth of what kind of microorganisms? Mesophiles, which are the ones that are pathogenic as well. And so really what we're doing here, yes, heat will improve the effectiveness of a chemical. I don't disagree with that. But in terms of the time of contact between what we have on our hands and the washing, there's probably not much of a difference. At least that's what I would argue. Everybody has to make their own decision on it, but it's pieces of evidence like this that I use to support that claim. I remember getting yelled at in 7th grade for washing dishes from some mom, I don't remember who, getting yelled for washing dishes with tap water, like regular room temperature water than hot water. And I'm not saying I'm vindictive, but I remember certain things like that, and I want to go tell her something, but leave it at that. All right. Questions on how to read these or how to analyze this data? All right. No, so thanks for asking. In my mind, if you were to superimpose these on each other, they would look identical. Mm-hmm. That to me says that because the results are very similar at 22 and 40, it says the temperature has no effect. Okay. If the temperature did have an effect, we'd expect to see probably, given how we've been taught to wash hands, that there would be a greater decrease here. Okay. Yeah, that's how I would look at it. All right. Who had to describe figure 3? Lexi, would you or Megan or Lauren be willing to tell us what this experiment is?
I suppose we can do that.
I would thank you.
So on the bottom, it's measuring like storage. Is that like how long it sits on your hand, like the silver?
This is in the test tube, actually.
Oh,
what basically similar setup to the last experiment, but now they're allowing the bacteria to sit in that soap for 0 to 72 hours.
All right, so it's like sitting on your hand. Well, whatever. Anyway, so it shows that the antibacterial soap, the population is 0 when it hits 24 hours, and then the plain soap is 0 at 72. So technically, antibacterial soap could be better, but no one washes their hands for that long, so I feel like neither is better.
Yeah, that's the point I always bring up, is who's going to sit there for 24 hours with their hands sitting in soap? And the answer is nobody, right? So if we look at it, and you've pointed out exactly what I would expect you to pick out, that 24 hours, the antibacterial soap, as you said, all the bacteria are gone at 72 hours with regular soap. What's happening in the first, or when do we start to see these diverge from each other?
After 12.
Yeah. Somewhere around 12, maybe, I think some people pointed out like 6 to 9 hours maybe, you might consider that to be a point at which you start to observe a difference. But even that is well beyond the amount of contact time that most of us have with soap and water. So there's a couple ways you could look at this. For the purposes of our own hand washing, does triclosan have an effect based on the first couple of experiments we've seen? No. When might something like this information be useful? So in other words, when are cases when you might have soap in contact with something for 24 hours? Grace?
Like equipment, medical equipment?
Absolutely. Medical equipment would be one place where you might find soap and water being used as a way to remove dirt, grime, degerm a surface. I don't know about you, but I have on more than one occasion left dishes in the kitchen sink overnight, and I usually put soap and water in them. It could be effective in those cases, right? If I were allowing it to sit for 12 or 24 hours. Okay. Any questions on this one? I think you hit the main point, Alexi, though. Who the hell washes their hands for 24 hours? And the answer is no one. No one does. All right. This last figure really gets to probably answering the hypothesis of maybe answering the main question. Does triclosan in hand soap have an effect on bacterial populations? Who had to, Alana, I think you and Anna and Taylor had to model this experiment, right? Can you just describe to us how they're performing this experiment?
So basically, they look at someone who doesn't wash their hands.
Uh-huh.
And they look at the bacterial population
That one.
That first one, so there's kind of a lot of bacteria in it. And then they look at someone who washed their hands with plain soap, and then they compare that to someone who washed their hands with antibacterial soap. And obviously, the bacterial population goes down, but there's not much of a difference between plain soap and antibacterial soap.
We'll come back to that last part in a minute. This is a highly controlled experiment, though, right? They're not just testing all bacteria that are on the individual's hands. Let's dive a little bit more into the actual experimental setup so that to allow them to control this experiment. How did they do this? Go ahead, Mom.
They had to put bacteria, specific
Yeah.
bacteria on there.
What bacteria did they put on the hands of the individuals? Serratia marcescens. There are two groups in here who had that as their unknown. You guys did, right? Mm-hmm. What color is Serratia?
Like pink.
Yeah, it's a pink reddish color. It's really easy to pick out from background noise. So the reason why they use something like Serratia here is because it's easy to distinguish from the natural bacteria that are on somebody's hands. Lindsay and Taylor, you guys had Serratia as well, right? So we can use this as like an indicator strain. So they basically forced people to put it onto their hands, wipe it on. What else did they control for? There's at least a couple of other things in this experiment that they controlled for to make sure that the only difference was the soap used. Annie?
They taught people the same way to wash hands.
They taught the individuals how to wash hands in the same way. What's the other thing that they did? Might not be that apparent, but I think they say something like they made sure to give everybody the same exact amount of soap as well, so that didn't have an effect. So using those controls here, then getting back to Alana's point, Alana, you said there's really not much of a difference. What are you using to support that conclusion? Good. So that's showing you basically the range of bacteria that was on the hands of the individuals they tested. Uh-huh. Yeah. Yeah, and I think those are all really good points. The points that I would bring up, if I were arguing there's no difference, these averages are pretty similar, and so are the medians, which I think are the dotted lines, if I'm not mistaken. These whiskers are actually within the same range, or at least cover the same range for both of them. And for the antibacterial soap, its range is completely within the range of the regular soap and water. Those would all be arguments. The term I would use is they're similar. Because they're similar, it indicates there's no difference between the two of them. Okay. Do both of them work better than just water? Yeah. Why? What is actually happening here? Degerming. This is a degerming process. The surfactants, the detergents that are found within soap are going to be used to mechanically remove the bacteria, mechanically remove dirt, grime, etc. from our hands, which will make it easier to remove the bacteria as well. Okay. We're not done just yet. Taylor, you guys had a question referring to, I think, if I'm not mistaken, I'll read it out real quick. So summarize the major findings and conclusions, and then given the results of the study and the potential effects of triclosan, should the FDA have banned its use from hand soaps? So in other words, you're the investigator at this point. What are the implications of this study?
We talked about that it should have been banned because the effects of triclosan, like the possibility of like the side effects we listed in the beginning, like those negative effects don't outweigh like the study that was like done where there wasn't much difference. If there's a bigger difference, then potentially that would have outweighed the negative effects that come with it.
Yeah, that's a really good point. So basically comparing the benefits versus the negative side effects, and here there really doesn't seem to be a benefit per se, at least in its intended usage. And so we would look at those negatives and say they're too great to outweigh the benefits. I think that's a great point. Other ones, does anybody have any other implications or conclusions from this study? So let me just summarize what's been done since this paper came out. Last year, about September or so, the FDA, I think it was September-ish, could have been earlier than that, the FDA banned 19 chemicals from antimicrobial soaps, or antibacterial soaps. Triclosan was the top one because that's the one that's been used most often. The only one that wasn't banned at the time was an ammonium chloride, so one of the quaternary ammonium compounds, because there hadn't been enough evidence yet to say that it did or did not have an effect. So the FDA was giving companies an extra year to show whether quaternary ammonium compounds had an added benefit in antimicrobial soaps. And I don't know what the ruling has been on that since then. Okay? All right. That is it for today. We got through the paper. Yay! So, we'll talk cancer. Question, Aliona.
For the exam, is it only one calculation possible? That you have to actually do?
Do you mean like type of calculation?
Like this one?
Yes.
Like just one of them?
I may have a couple of them on there, or it might be a slight modification of it, but it would be that type of problem. And if you want practice with those, there are a number of them up on Blackboard available for you guys to look at as well. And if you want more, I'll give you more. I've got plenty. There should be one on each of the exams too, if you
Okay.
want to look.